Hi everyone, and welcome to the 11th TIA Centre seminar of the academic year. My name is Adam Shepherd, and I'm a researcher here at the TIA Centre. For those that are new to our seminars, we invite researchers and leaders in the area of computational pathology and beyond to discuss their work. I'm also joined by our co-host, Jakshi, over there, who helps to organise and run these seminars. And it's a real pleasure today to introduce Mart van Rijthoven. Mart did his PhD work in the computational pathology group at Radboud University Medical Centre, focusing on deep learning for biomarker quantification in whole slide images. His work includes the HookNet model, quantification of tertiary lymphoid structures, and few-shot learning for histopathology. He also developed the whole slide data package and co-organised the Tiger Challenge. And the title of Mart's talk today is Analysis of Computational Tumor-Infiltrating Lymphocytes in Breast Cancer from the Results of the Tiger Challenge. So with that, Mart, I'll pass over to you. And yeah, thanks again for joining us.
All right, thank you so much. Yes, thank you for the introduction, Adam. I also prepared a slide for people that didn't know me, but actually you already introduced me very nicely. Maybe to add, so I was part of the Radboud UMC group in Nijmegen, the Netherlands. I was part of the diagnostic image analysis group, and in particularly the computational pathology group. And I was honored by being supervised by Francesco Ciompi and Jeroen van der Laak. And as Adam already told, I worked mainly on segmentation and detection, but also did a bit of work on future learning. And I was the co-organizer of the Tiger Challenge and author of the paper that we produced from it, analysis of computational tumor-infiltrating lymphocytes in breast cancer from the results of the Tiger Challenge. And my talk today is going to be about that challenge and also all the work that we did post-challenge to get the paper out. So first off, the Tiger Consortium here, it's a big list of people that worked a lot on the work that I'm going to present today. So it's definitely not a solo work from myself, but really a lot of people were involved and a lot of people put a lot of effort in this work. So already thanks a lot for all the people. Even also TIA Centre themselves were a participant in this challenge, and you will see also their results actually coming by later this presentation. So a bit of the overview of the presentation. First, I'm going to talk a bit about tumor-infiltrating lymphocytes. Then I'm going to present the Tiger Challenge, the Tiger Challenge results. And then I'm going to talk about the post-challenge analysis and work that we did, and then the results of that. And then in the end, I'm going to give a short conclusion about this work. So first, TILs. So why do we actually want to work on TILs? Well, clinical studies have already shown that it is associated with the patient response in immunotherapy and chemotherapy. Also, they have been shown to be predictive for cancer recurrence and patient survival in TNBC. And also they show a strong correlation with the PCR in TNBC and HER2. So there is actually quite some clinical studies that already show that the TIL is a very nice biomarker. So a bit, what are TILs then? So here you see an image of a part of an whole slide image, and here you can see the lymphocytes. And often lymphocytes and plasma cells are taken together as TILs because plasma cells are a bit difficult to distinguish in H&E slides from lymphocytes. And then we have the International TILs Working Group that made recommendations on how to score TILs in H&E. And here on the left, you see a slide. And the recommendations are a bit more complex, but the gist of it is that you first define a tumor bulk or a tumor bed, as shown here on the left. And then in that region where all the tumor sits, you're going to look for lymphocytes. And on the right, you see a bit of a zoomed in region. So you only look in the area within the tumor borders for these lymphocytes. And there are actually multiple ways to compute tails. There are also kind of variations of tails. So you can have the stromal tails, and the stromal tails are basically the lymphocytes that are in the tumor bed or in the tumor bulk, but that are in the stroma. And so then you compute basically the ratio of the area covered by the lymphocytes within the stroma. But you can also have like intratumoral tails. And then you're going to look at really the tumor cells and the lymphocytes that are located there. And then you also have peritumoral tails, and peritumoral tails are actually a bit different because they also, then you look at the borders of the tumor bulk. So you don't take into consideration the full area here, but you only look at the tumor border of the tumor bulk, and you only take that into consideration. So there are multiple ways, and actually the International Working Group recommendations have even more basically guidelines on how to process them. But for now, I think this will give you a bit of a clear view of how these are scored via manual way, in a manual way. So, and the problem is a bit here, it's a bit of an illustration where the problem actually occurs. Here on the top, you see like a illustration that it is probably quite easy to estimate the percentage of the colored or team players on the benches. That's quite easy to do for a human. If you ask multiple humans, they probably come up with a pretty same percentage score. But if you look at the bottom illustration, if all these persons with all the team member shirts are kind of hustled, it's very difficult to give a percentage to how many yellow team players there are, or purple or red. It's very difficult, and even if you are an expert pathologist that have a lot of experience, this still is a bit complicated because it's just inherently difficult for human beings to do this percentage on these kind of complex things. So now back to these all slide images. This is of course a bit of a similar pattern that it's not that clear, and if you want to put a percentage somewhere, it's quite difficult. And if you ask multiple pathologists, they often give different scores to the same slide. And well, for a full slide, it's even impossible to give the right answer. But even if you look into specific regions, the choice of the regions that you're going to look at might be different from one pathologist to another one. So to overcome this and have a bit of reproducible results, we want to introduce basically an automated way to compute these tumor-infiltrating lymphocytes with AI. So that's basically also the question that we ask ourselves for this paper. Like, can we accurately quantify these computational tails in breast cancer? And then we want to check if we can get strong correlation with visual pathologist assessments. We want to check if we can predict pathological complete response, and if these computational tails can provide independent prognostic value for patient survival. So first, then we actually launched the Tiger Challenge. So Grand Challenge is a website. I think most of you know this website, but on this website you can host well, basically medical challenges, and a lot of kind of successful challenges already have been deployed there. And we thought that by also deploying this challenge here, we could actually gain from the community basically really good algorithms instead of just trying to make our own algorithm. We wanted to see if we ask a lot of people internationally, if we could come up with a lot of good algorithms. And how we did this is called a type 2 challenge because we had actually data that we could not share with all the participants because they were, we were not allowed because of patient data. So a type 2 challenge works as follows. You have the participants environment, and then we have the Coin Challenge platform. And for the participants environment, they can actually download the training data. This challenge was also sponsored by AWS, so we could put all the training data on AWS, and then participants could download the data to their local environment or to their cluster and do the model development themselves. Then, when the participants were happy with the model, they were asked to make a Docker container, an algorithm container, and then this Docker container could be uploaded to the Coin Challenge platform. And then on this platform, where we have the sequestered test data sets, we can run the algorithm on this test data and evaluate the algorithms. And this way, we could actually use data from patients that we could not share publicly, but still have the algorithms run on it. And I think this is a really nice setup, although it comes also with a bit of complexity because, of course, you have to upload the Docker, and then the Docker will run on this challenge. And yeah, probably often this will not go in one go, so you will get errors, and then you have to give feedback to the participants. And well, at this point, I also want to say that it was really nice to work with all the participants in the challenge because they were very patient with receiving the feedback from the errors that we collected on Coin Challenge, and then they had to modify their Docker container in their local environment, and it was quite of a complex way of working, but in the end it worked out very well. So that was really great to see that a lot of participants were really great in this collaboration. So that was really nice. So here you see a bit of the overview of the Tiger Challenge timeline. So first we had the training data released, and then we had a period of the development of the AI algorithms. So basically we have two tracks. So we have the computer vision track and the survival track. And then we had also an experimental phase and a final phase. So in the computer vision track, participants could upload the Docker, and then the algorithm was evaluated on tissue segmentation and lymphocyte detection. And for the survival track, we actually ran the survival analysis. And this, participants could upload multiple Dockers multiple times per week to basically fine-tune their model as a validation phase, basically. And then in the end, you had the final phase for each track, and then the participants were only allowed to only choose one of their Dockers they made, and we would run their algorithm only one time, and that would be then the final answer, basically. So basically here a bit of a same story that I already talked about, but we have the Tiger training data, which was it included data from RoboTMC, Jules Baudet, and TCGA. And then the participants should do tissue segmentation and lymphocyte detection, and from that compute a TAILS score. However, it was actually also allowed to only submit Docker to the survival track, So then you didn't have to do the tissue segmentation and lymphocyte detection, and also the other way around. You were also allowed to only submit to the computer vision track. And actually, we will see that 2 teams actually did that. Now, for the training data, the training data actually consisted of 3 different types of data. So we had whole slide image ROIs, which contained of region of interest in whole slide images containing the ground truth segmentation and the lymphocyte detection. So here you could really train a model for segmentation and detection. But then we also had a WSI bulk data set. This is basically the bulk which I was talking about in the beginning for the recommendations by the TILS working group, that you want to basically specify first the whole set image bulk where you want to do your analysis for your TILS. So you could basically also use this in your training pipeline to basically find out where the tumor bulk is. And then we had also a set of WSI TILS, which were also images that were scored by a pathologist that would give a TIL score to the full slide. And this you could use for maybe calibrating your TIL score, or maybe fine-tuning your TIL score after you produced it from the segmentation and detection output of your models. Then we have the data, of course. Of course, then we need the participants to make the challenge successful. And here you see an overview of all the participants that signed up. There were a lot of people that signed up, but actually did not really actively participated. But from a lot of countries, people were interested in the challenge. But in the end, we had 45 teams. And these are the submissions. And so for the computer vision, we had 329 submissions. And for the experimental survival, we had 49 submissions. But these were, of course, then multiple submissions of the same team. For the final leaderboard, you could only submit once. So from the 45 teams, only 11 submitted for the final computer vision. And for the final survival, only 9 teams submitted. And this also indicates a bit the complexity of the problem that we were trying to solve, because I think a lot of people were interested, and also 45 teams started working on it. But also to have a successful algorithm running with reasonable results, it was actually quite a challenge. But in the end, we had some great teams that submitted some very nice algorithms. And here you see the computer vision final leaderboard from the current challenge. So this was basically the final result of the challenge for the computer vision track. And here you see that Biototem, Tiger, and AIVIS were the top 3. Tiger is a team from the TIA Center, so that's pretty cool that they were second place here. So we evaluated the tumor stroma dyes and the FRC score for lymphocytes. And then we took the ranking, we took the mean ranking of these both of these metrics, and that determined the final position in the leaderboard. And yeah, these algorithms really produced very nice segmentation and lymphocyte detection. Then for the survival leaderboard, we used the UNOS AUC. Actually, during the challenge, we called it the C index. That was not completely correct. UNOS AUC is also a time-dependent metric. It's very similar to the C index. That's why we called it in the challenge the C index. But we also provided all the code for the evaluations online. And there you can also see that the actual procedure was the UNOS AUC. But the C index would give very similar results. The winner of the challenge was Vuno. Very interesting. We will see also later on their way to compute CTILs. Dropout UMC was a baseline model developed by a master student, Cyril. But yeah, because we were the organizers and the baseline model was only for basically, it wasn't really a result that would participate in the final leaderboard. So it was not counted as a real position. So spotlight pathology and cells vision were second and third. Interesting here is that, Tiger and Biototem scored a bit lower, while they were actually first place and second place in the computer vision leaderboard. And the reason we have some ideas why, but actually later in the post-challenge result, we see actually that their algorithms are actually also producing quite nice results. And it could just be that the data set where they were just a bit unlucky here. But we will get into that a bit more later. So from all the teams that submitted to the computer vision leaderboard and the survival leaderboard, we took the top 3 computer vision teams and all the survival teams that submitted to the survival leaderboard. And here's a bit of an overview of what the teams did based on segmentation and detection and how they computed the tail score. In the supplement that we're going to publish also, there will be a very extensive method description for each of them. So if you want to know more details, that will be available later. But to summarize, actually all of them used CNN-based models. Of course, this was also done in 2022. So it's the transformer architecture was not that popular for these kind of tasks at that moment. As you can see here, that SRI actually didn't do the segmentation and detection. They didn't they didn't make a Docker for the computer vision track. And IFS didn't make a Docker for the survival track. That's why we don't have any information here. Interesting that Funo was the only one that did the peritumoral tails. That was the tails version that I was talking about in the beginning of the presentation about the border of the tumor bulk. And the other ones mostly did the stromal tails. And one reason could be that Tiger and Biototem, maybe they did basically the amount of border of the extent of the border might have influenced their scores in the survival leaderboard. But as we will see also in the post-challenge analysis, they are actually scoring pretty similar as the other models. So here you see an overview. The upper part I already presented. There was for the model development, and now for the model benchmarking, we extended all the data for the post-challenge as well. And here you can see a bit of an overview of what kind of analysis we did. So we did computer vision performance, survival on sections, correlation with pathologists, PCR prediction, and survival on biopsies. And then here we have the annotations available for each data set. So basically these two, the detection tiger and segmentation tiger, these were from the challenge. And this data set we added. I will talk about it in the next slide. And then here also part of it is from the challenge itself, but then we also extended it with a lot of extra data to do some extra analysis for the algorithms. Yeah, so first thing what we did was for the extended computer vision analysis, we used immunohistochemistry to create the ground truth for a couple of slides. So we did destaining and restaining for the same slide, and then did some registration for the immunohistochemistry stain and the H&E stain. And what we could do then is, based on the immunohistochemistry, you could identify the lymphocytes in a more, let's say, robust and way, and then match them on the H&E slide. And this actually allowed us to compare basically what the results from the challenge that were actually made manually on H&E. And here you see basically the final computer vision performance. On the right, you see the dice scores for stroma dice and a tumor dice. Basically, these are the metrics that we also obtained during the challenge. So as you can see here, that IFIS, Biototem, and Tiger were at the top, the top results. Interestingly, is that you can see that tumor dice and stroma dice are a bit similar. You see that the stroma dice is a bit easier, it seems. And then for the FRC scores, the detection tiger, so on the x-axis, these are basically the scores that we obtained during the challenge, but then we also added the restain detection, and then we could check how well that actually performed. And well, we can see actually that it is pretty okay. You see a bit better performance on the detection tiger, but I think it really shows that the ground truth for the detections in the challenge were pretty good. So that's a nice result. One thing to note here is Cells Vision. That team scored a bit less, but we will see actually that they are doing pretty well on the survival leaderboard. And the reason why they are doing not so well is not because they have a bad model, but also looking at the results they had, something went wrong in the processing. So for the dice scores and the FRC, the participants were asked to process region of interest within the whole slide image. So not the full whole slide image, but regions that we defined. And something went wrong in that processing for Cells Vision. So actually their dice score and FRC scores are not really good representing their quality of their segmentation and detection results, unfortunately. But that's an extra mark, because we will see that Cells Vision is actually doing pretty well also. So the next result is actually the correlations with the visual tails. We had quite some extra data sets added after the challenge, where we had visual tails, so tails scored by a pathologist. And we see actually that for some data sets, it's doing pretty well. And for other data sets, it's not going that well. Also interesting to see is that Vuno scored a bit less here. And actually that makes kind of sense, because they did the the peritumoral tails, while the visual tails scored by the pathologists were all stroma tails. So it makes sense that they are a bit different. And here are the results for PCR. So we calculated the area under the curve. And for all methods, we see actually that for TNBC, they are doing pretty well. We also have the V-tails, so the visual tails from the pathologist. And as we see that the algorithms are actually really close to the values of the V-tails, so indicating that for TNBC, these C-tails methods actually perform similar as the pathologist. And for HER2, we actually see that some are similar, but also some are even better. So these results were really nice to see, that for PCR, these C-tails methods could actually be very useful to automate PCR prediction. Then Then we did prognostic value in univariable analysis. And here you see two Kaplan-Meier curves. I only took two of them based on the p-value. So these methods scored the lowest p-value for right about for TNBC, the disease-free interval, and Tiger for TNBC overall survival. So very interesting actually that Tiger is in this post-challenge data set on resections. They actually performed well based on the p-value best in overall survival. But basically all methods performed similar on TNBC. So this was a very nice result, also showing that these CTILS methods are really able to make these two groups. These groups were defined on the median TILS values for each. Yeah, so that was a nice result. If we go to the HER2 plus data set for resections, we actually see that the results were a bit, well, were not that nice. What we see here is also, again, based on the lowest p-value, but these values are not even significant. And this was also across actually all CTILS methods, that for HER2 plus, we didn't really see this separation as nicely as in TNBC. Then we did also Cox regression model. And here actually you see also again, basically the same story for DFI and OS, that all methods really have nice results for hazard ratio and confidence intervals that are all very highly significant for TNBC, but for HER2 plus, not the same result actually obtained. Then we also did multivariable analysis, and here you see the C index. And what is really nice to see here is that we also did the clinical model, of course, and then we can see that all methods actually improved on the baseline model. So all of them have added value on top of the baseline covariates. Really nice. But also on HER2, you see not the same beautiful result. You see a bit that the some methods are have added value, but some are not. So it's really yeah, inconclusive. Then also, if we look at the multivariable results in the Cox models, hazard ratios, the TNBC look pretty nice. Also, also significant. And also, again, in the HER2 subset, we don't see the same results. So that's really a recurring thing. Very interesting because the on the PCR, HER2 was successful. Then for biopsies, well, we had a lot of the data sets for biopsies. But also here, we don't really see a consistent result for TNBC, but also not for well, here we see when the, oh yeah, wait, before I forget, for the biopsies, we actually also have visual TILs. So we could also measure the hazard ratios for the visual TILs. But we see actually that also for the visual TILs, often the results are not really what we would like to have. Sometimes we have here a significant result, but then the models are not significant. And here also, and here as well, where in G7 we have a bit more significant results for the CTIL model, but it's not really consistent. And the same story for HER2 plus. Also, again, not really the results that we got from the resections, actually. I'm already at the conclusion. So based on these results, we could say that algorithm design matters. So really defining the tumor bulk and how wide you have the tumor border of this tumor bulk actually impacts the clinical performance. Computer vision versus clinical impact. We have seen that some CTLS methods have very good computer vision scores, but did not really have survival prediction in particular subsets. In other subsets, they actually were performing well. But it's not really like if you have computer vision scores that are great, that you will have a nice survival prediction. And actually, what we see actually is that even if you have computer vision scores that are not perfect, you can actually do quite well in survival prediction. For treatment response, we have seen that CTLs predict PCR in most data sets for both TNBC and HER2+. So that was really a positive, a nice result that we showed. For prognostic value in resections, we have shown that CTLs show significant prognostic value in resected TNBC, but we didn't see the same in HER2+. And for prognostic value in biopsies, yeah, we actually saw that CTLS lacks significant and consistent prognostic value across biopsy cohorts. One reason could also be that for biopsies, it's a bit more difficult to define the tumor bulk. For resection, it's often easier. So that could also be a reason. Another reason is that could be that the biopsy data sets were actually also for the visual tails not significant. So it could also be that the data sets were not really didn't really show any prognostic value at all. So then the future of Tiger, very interesting. I'm not at Radboud UMC anymore, but at Radboud UMC, Francesco Ciompi and also Roberto Salgado are using these Tiger algorithms to apply them on even more data. So that's a really cool thing, and probably we will see some results soon. And when the paper will be published, we're going to transition from basically the challenge to a continuous open benchmark for the community. So what we're going to do is we're going to reopen the challenge, but not for the challenge itself, but where people just can upload their algorithms and test their algorithms and see the results. And we are also planning to open it in a way that it's both comparable to the challenge results, but also partly to the results that we have, that we are going to present in the paper, probably for the TNBC cohorts where it really worked well, but that we're going to discuss further soon. Then, of course, thank you for all the participants and all the people that were in the consortium, but also to a lot of institutes and hospitals that were involved, also to Grand Challenge and AWS as a sponsor. A lot of people were involved, so thanks to for all of them. Yeah. I think that was my presentation. Still have 15 minutes left, so I think nice time to go to questions.
Fantastic. Well, thank you very much, Mark, for a really, really interesting talk. And again, congratulations for organising such a great challenge, and for, of course, having the publication as well very recently. So yeah, let's open the floor to questions. Does anyone have any questions online or in person they'd like to kick us off with? I'm sorry. Yeah, of course, actually. Feel free to.
Hi, Mark. I'm Jachi. So I have firstly, that's a really good paper, and actually I worked on Tiger after the challenge is finished. I tried to work on it a little bit. I don't know if you remember my, but yeah. So I have a few questions. First, also, Yeah, so in this work, a lot of teams calculate the TILS score based on this defining the tumor bulk first. Do you think that's one of the issues that causes some sort of, I don't know, a mismatch between computational methods and human kind of interpretation of the tumor bulk? And if for people that's working on this computational scoring today, would you still recommend using this tumor bulk region, or would you move towards a kind of a learned spatial aggregation strategy to
Yeah.
generate?
Yeah, it's a good question. I think actually more research is needed to really see what the effects are actually for these regions. We have seen indeed that actually I think yeah, self-sufficient did a more aggregation from different regions, which actually also worked well. So I think it's a really active part of research that needs to be done. What I've seen doing my own experiments is that defining the, let's say, border of the tumor bulk, and if you extend it a bit more or a bit less, then actually it can impact the survival predictions quite well. And it actually also makes sense because sometimes there are a lot of lymphocytes at the border, and if you include them or not include them, that will definitely change your CTIL score, and that will impact your predictions. I do think that the recommendations from the TILS working group, they really have thought about it. So trying to basically make an algorithm that at least adheres to those recommendations a bit is a good strategy. But also other strategies can be explored. But I wouldn't say don't use these visual recommendations at all anymore. But I think I also don't think that there will be one answer. So more, I would say more research needs to be done there to really find out what the effects are. But it is also complicated because you're also restricted to the data sets that you are working with. So in the end, you want to basically have a even larger data set where, yeah, the data set size and other biases are not in play anymore, and you can really focus on the actual process of determining TILs instead of the extra additional biases that you introduce. So yeah, it's an open question, I would say.
Yeah, okay. Yeah, thank you. And one more question about your results. I think I remember there was one cohort that's called G8 that was scored had really low correlation. Why is that?
Yeah, G8 was data set from the German breast cancer group, and they did the analysis for us because they were not allowed to share all the patient data. So we couldn't really, I didn't really explore it in depth because we didn't have the raw survival data. So there could be many reasons, but what we have seen actually is that for G8, a lot of analysis were not that great. So I would actually say that G8 was a data set that was just not really performing well because it didn't really include these prognostic information that we were searching for. But to really make that hard, we should look at the clinical data. But we didn't have access to that. Could be something that we could explore with the GBG later on. Yeah.
Okay, thank you.
Brilliant, thank you. And just yeah, I've got a couple of questions sort of to follow up on that really. So I don't know what was the if there's any sort of scientific rationale or thought about in general seeing much better sort of predictions for the triple negative cases over the HER2. Is that something that you expected to see, or?
Well, there is a bit more evidence, I would say, for TNBC. Also, one could say that morphology in the morphology of TNBC, it might be a bit more easier. Though I don't have really a medical background, so I'm a bit. I don't want to say too much about the biological explanation about it. But another reason could be that actually in the training data, also more TNBC cases were included than HER2. So that could also be a bias to the CTILS models. But yeah, that would be yeah, that could be.
Sure, thank you. And also going back to the slide, of course, where you showed the results, and I think, yeah, please correct me if I get your explanation slightly wrong here. So in terms of what we saw in terms of the methods that did really well for segmentation, detection, and sort of finding these TILs, but then almost had there was sort of the reverse performance on the final leaderboard for the sort of the final C index or the AOC. I know you said it's on sort of later testing that hasn't necessarily been the case, but you think it's just a case of sort of being unlucky on that final test set? Because it was still quite a big test set, right? It was about 700 cases or so.
Yeah, true. But what we actually also saw that, so maybe to make that clear, the resections, TNBC, that we have now in the paper, these are basically the TNBC cases that we also had in the challenge. And we added more HER2 cases. And then we stratified TNBC and HER2 in the paper, which we didn't do during the challenge. So the challenge data set had TNBC and HER2 in the same data set. And it might be actually that because we see that that your algorithm, for example, did pretty well also on the TNBC cohort in the paper, that the HER2 examples, or sorry, the HER2 slides in the challenge data set were actually the cause of the less good result that we observed. Then in the paper where we stratify between TNBC and HER2, you see actually that your model is performing as well as all the other models. So yeah.
Sure. Okay. That's good to know.
Yeah, indeed.
Thank you. And do we have any more questions for anyone in person? Sorry. I suppose I might ask a quick question about in terms of actually organising this challenge, because it was obviously a massive undertaking, and you've had something like, I think you said nearly 400, more than 400 people involved in terms of submissions. So I just wondered if you could sort of tell us a bit more about this in terms of what was the hardest thing about organising this challenge, and what was the best thing too.
Yeah. So so organizing challenge is it's just a lot of work because so you have to. Well, it's actually a good thing because you have to be really like prepare a lot of stuff before you can even develop the models, which is actually a good thing. Sometimes if you work on your own research, you might go developing models before your data is completely clear. So, but having done all that is quite a challenge. Collecting all the data, collecting all the annotations. We actually made a relatively big data set for training, but also for the validation. And we made extra annotations with pathologists, which took quite some effort. Of course, we also used a lot of public annotations for the training. But building the whole data set was quite a challenge. And then, of course, you have we created a baseline algorithm, but also a Tiger example algorithm, which was also quite some work to make it general in the sense that everybody should be able to use it. And then we had some well, complexities on the Grand Challenge platform. So there were some limitations running the algorithm. There was a time limit. So all these things complicated a bit, but it was in the end really nice that it worked out. Yeah, so then I think the challenge went all right. I think one thing that I really liked about it was that we also shared all the code. So that is actually also a thing that all the code that you made is a lot of effort. But then if you share it, other people can also see it and maybe find some mistakes, which actually Spotlight Pathology did in the evaluation. So that was really great. And then, yeah, post-challenge, we wanted to run all the algorithms on the extra data sets, and that was actually also a challenge on itself. But then we were really in luck with all the teams because I might have expected that some teams would say, yeah, the challenge is over, bye. I'm going to work on something else. But every team actually was really motivated to work on this post-challenge data sets. So that was really nice. Yeah, I would do it again, but I would plan probably more time for it because it's just a lot of time. It just takes a lot of time. And I started doing this challenge in my, well, almost just before my last year, I guess. I would start it maybe a bit earlier to prepare everything because yeah, it just takes a lot of time. Yeah.
Absolutely. And if and today with the sort of if you're redoing this challenge today in sort of 2026 with all the sort of these foundation models that we have now, is there anything that you would change about the challenge?
Well, let me think a bit about it. If I would change something, not sure, but to be honest, I'm not super aware of the current state of the art of, let's say, foundation models for pathology at the moment. I know that there are some foundation models. One could, of course, use those as basically a baseline of a start, like a start. But that's actually the good thing that we're gonna open the challenge again, and then we can actually run these foundation models on this benchmark and see how well they are performing. But other than that, I think no, because yeah, the same story goes for the data collection, etc. That actually takes a lot of time, and that I would probably do the same. Yeah.
Thank you. Okay, then I think we've probably got time for one final question. If anyone online or in person's got a question I'd add, I know someone had wrote a message, but it looks like they've deleted it now, so might have changed their mind about that question. But if not, we will draw the seminar to a close then. Mart, thank you very much again for joining us and for giving us such an excellent talk for discussing the Tiger Challenge and the results. And congratulations again on a successful organisation and getting the paper too. So thank you.
Thank you so much.
And a reminder to everyone else that our next seminar is scheduled for the 23rd of March, where we're going to be joined by Dr. Johannes Lotz from Mevis, where he's going to discuss some of his lab's work and also some of the results actually on the Unicorn Challenge. So thanks everyone, and thanks again, Mark, for joining us.
All right, thank you.
Bye everyone.
Bye bye.